Blood is a crucial component of our body that helps us stay alive by transporting oxygen, nutrients, and hormones to various parts of the body. It also helps remove waste products such as carbon dioxide and other toxins. The blood in our bodies contains a variety of different cells, including red blood cells, white blood cells, and platelets. However, what many people may not realize is that there are also different blood types, which are classified based on the presence or absence of certain proteins on the surface of red blood cells. In this video, we will explore the reasons why we have different blood types, how they are inherited, and their significance in medicine and blood transfusions. The discovery of blood types. The discovery of blood types is credited to an Austrian physician named Karl Landsteiner, who made the discovery in 1901. Landsteiner noticed that when he mixed the blood of different individuals, it sometimes resulted in the formation of clumps or agglutination. He identified 3 different types of agglutination, which he called type A, type B, and type C. Today, type C is known as type O. After discovering the different blood types, Landsteiner also found that each person's blood could be classified as one of these 4 types, based on the presence or absence of certain proteins on the surface of their red blood cells. These proteins are called antigens, and they are what cause the agglutination when mixed with certain blood types. Blood types and antigens. There are 2 main types of antigens that determine blood type: ABO antigens and Rh antigens. ABO antigens. The ABO antigens are the most well-known and important antigens in determining blood type. These antigens are found on the surface of red blood cells and are designated as A or B. If a person has the A antigen, they have type A blood, and if they have the B antigen, they have type B blood. If they have both A and B antigens, they have type AB blood, and if they have neither, they have type O blood. Rh antigens. The Rh antigens are another type of antigen that can be found on the surface of red blood cells. They are either present, Rh+, or absent, Rh, in an individual's blood. People who have Rh+ blood are said to be Rh positive, and those who do not have the antigen are Rh negative. How blood types are inherited. Blood type inheritance follows the principles of Mendelian genetics. Each person has 2 copies of the ABO gene, one inherited from each parent. The gene has 3 possible variations, A, B, and O. If both parents have the same blood type, their children will inherit the same blood type. For example, if both parents have type A blood, all of their children will also have type A blood. However, if both parents have different blood types, the child's blood type will depend on which gene they inherit from each parent. If one parent has type A blood and the other has type B blood, their child could inherit either the A or B gene from each parent. This would result in the child having either type A, type B, or type AB blood. If both parents have type O blood, all of their children will also have type O blood, as both parents only have the O gene to pass down. The inheritance of the Rh factor follows a similar pattern. If both parents are Rh positive, their child has a 75% chance of being Rh positive and a 25% chance of being Rh negative. If one parent is Rh positive and the other is Rh negative, the child has a 50% chance of being Rh positive and a 50% chance of being Rh negative. If both parents are Rh negative, their child will also be Rh negative. Significance of blood types in medicine. Knowing a person's blood type is crucial in medicine, particularly in blood transfusions. If a person receives the wrong blood type during a transfusion, it can cause a potentially fatal reaction. This is because the recipient's immune system will recognize the transfused blood as foreign and attack it, causing agglutination and damage to the red blood cells. For example, if a person with type A blood receives type B blood, their immune system will recognize the B antigens as foreign and attack them, causing agglutination and potentially life-threatening complications. Therefore, before a blood transfusion, the donor's blood type must be matched with the recipient's blood type to ensure compatibility. A person with type A blood can receive blood from a type A or type O donor, but not from a type B or type AB donor. Similarly, a person with type B blood can receive blood from a type B or type O donor, but not from a type A or type AB donor. A person with type AB blood can receive blood from any blood type, while a person with type O blood can only receive blood from a type O donor. The Rh factor also plays a role in blood transfusions. A person who is Rh negative can only receive blood from an Rh negative donor, while a person who is Rh positive can receive blood from either an Rh positive or Rh negative donor. Blood types also have implications in pregnancy. If a woman is Rh negative and her partner is Rh positive, there is a risk of hemolytic disease of the newborn, HDN, where the mother's immune system attacks the fetus's red blood cells. This can cause anemia, jaundice, and potentially life-threatening complications. However, this can be prevented with Rh immunoglobulin injections during pregnancy and after childbirth. Conclusion. In summary, blood types are determined by the presence or absence of certain antigens on the surface of red blood cells. The ABO antigens and Rh antigens are the most important antigens in determining blood type, and their inheritance follows the principles of Mendelian genetics. Blood type compatibility is crucial in medicine, particularly in blood transfusions, to prevent potentially life-threatening complications. Knowing a person's blood type can also have implications in pregnancy and childbirth. Overall, understanding blood types and their significance in medicine is essential for healthcare professionals and the general public.